Hello everyone, my name is Sarah Francis Wani, student at Industry Hungary, Level 3 Biomedical Science. I'm going to present about Elisa technique, difference between sandwich and indirect, application of Elisa in clinical practice, the types of vaccines and their characteristics, and then how does herd immunity contribute to disease prevention, then the difference between active and passive immunization. By starting with the principle of Elisa technique, Elisa is enzyme-linked immunosorbent assay. This is a technique widely used in laboratory method to detect and quantify substances such as proteins, hormones, antibodies, and antigens. The basic principle of ELISA is based on the specific interaction between an antigen and its corresponding antibody. How does it work? First of all, there's a specific binding whereby the antibodies bind specifically to antigens and vice versa. We have enzyme reaction. The enzyme linked to the antibodies produce a detectable signal, the color change or fluorescence. We have quantification. The signal is measured and correlated to the concentration of the target. We go to the second question. The second question says, how is the sandwich ELISA different from an indirect ELISA? The sandwich ELISA detects antigens like proteins, but for indirect detects antibodies like IgG, IgM. Then two antibodies capture and detection for the sandwich, and then for the indirect, one primary antibody and secondary antibody. Then for the sandwich again, antigen is sandwiched between two antibodies, but for indirect, primary antibodies bind to the antigen, then secondary antibodies bind to primary antibody. Then for the sandwich, high sensitivity due to two antibodies binding, and then for indirect, moderate sensitivity depends on primary antibody. We go to the next, which says the applications of ELISA in clinical practice. First of all, the diagnosis of infectious diseases, like detection of antibodies. ELISA is commonly used to detect antibodies in a patient's blood, which are produced in response to infections. Like the diseases we have, like HIV, stroke, AIDS. This detects HIV antibodies or antigen. We have hepatitis, whereby the ELISA can detect hepatitis B and C antibodies or antigens such as HBsAg in hepatitis B. We have the Lyme disease, detects antibodies against Borrelia. We have malaria. ELISA detects antibodies or antigen for diagnosing malaria. The second application, we have humoral assays. The ELISA is frequently used to measure the concentration of various hormones in blood, including the thyroid hormones. Then we have the human chronicle, like the HCG used in pregnancy test to detect HCG in urine or serum. We have insulin glucose. ELISA can be used to monitor insulin levels in patients with diabetes or to detect insulin resistance. We have cortisol, measuring cortisol levels to diagnose adrenal disorders such as Cushing's syndrome or Addison's disease. Then we have detection of autoimmune disorders. ELISA is often used to measure antibodies in patients suspected of having autoimmune diseases such as the rheumatic arthritis, then celiac disease, cancer. Then we have the cancer marker detection. ELISA can be used to detect tumor markers that indicate the presence of cancer or monitor treatment response, like the prostate cancer, the ovarian cancer, breast cancer, liver cancer, etc. Monitoring inflammation and infection. The CRP, like we have the CRP, is used to measure CRP levels which rise during inflammation and infection, making it useful biomarker in the monitoring of conditions like the cardiovascular diseases. Then we have food and drug allergy testing. IgE testing, like ELISA can detect specific IgE antibodies in response to allergies, e.g. like peanuts, eggs, shellfish. This is useful for diagnosing food allergies. Then we have drug hypersensitivity. It can also be used to detect antibodies related to drug allergies or drug-induced immune reactions. We have blood and organ transplantation. Cross-matching and organ transplantation. ELISA is used for organ transplantation to detect antibodies against donor antigens, e.g. in kidney, liver, or heart transplants, and assess the risk of rejection. We have detection of HLA antibodies. These ELISA can be used to assess compatibility between organs, donors, and recipients by detecting HLA antibodies. Screening for genetic disorders. ELISA can be used to detect biomarkers that may indicate genetic disorders. We also have the therapeutic drug monitoring. ELISA is used to measure drug levels in the blood to ensure proper dosing and avoid toxicity. We have environmental and occupational health. ELISA is used to detect toxics and pollutants that may affect human health. Our next question, we have the types of vaccines and their characteristics. The first one, we have inactivated killed vaccines like the polio vaccine, hepatitis vaccine, hepatitis A vaccine, rabies vaccines. Their characteristics are like they are safe for immunocompromised individuals since the pathogen is inactivated. Usually required multiple doses to achieve long-lasting immunity. Often combined with adjuvants and then like substances that ensure the immune response. Limited immune response compared to live vaccine. Then the second, we have live attenuated vaccines. For example, the measles vaccine, chickenpox vaccine, rotavirus vaccine, yellow fever vaccine. The characteristics, we have strong and long-lasting immune response, often requiring only one dose. It may be unsuitable for immunocompromised individuals like people with weakened immune systems because the attenuated vaccine can potentially cause disease in these individuals. Then fewer doses and less frequent boosters are typically needed compared to inactivated vaccines. The third vaccine, we have the subunit, combinant, conjugate, and polysaccharide vaccines. These vaccines are fragments of the pathogens such as protein, sugars, or parts of the microorganisms like the outer coat proteins to stimulate an immune response. The pathogen itself is not used, reducing the risk of side effects. The subunit vaccines contain only a specific piece of the pathogen, e.g. protein or sugar. Then the recombinant vaccines use genetically engineered microorganisms to produce antigens, e.g. the proteins from the pathogen. Conjugated vaccines combine a weak antigen with a stronger antigen to improve immune response, e.g. attacking bacterial sugars to a protein. Polysaccharide vaccines, they contain long-chain sugar molecule from the surface of protein. And from this type of vaccine, we have like the examples, we have the hepatitis B vaccine, HPV vaccine, and then the characteristics, they are safe for immunocompromised individuals because they don't contain live pathogens. They are weaker immune response compared to live vaccines, so they may require boosters or the addition of adjuvants. High specific in targeting parts of the pathogen, reducing the chance of side effects. Toxoid vaccines. These vaccines contain inactivated toxins produced by bacteria. The bacteria's toxins are inactivated so they can no longer cause harm but still stimulate an immune system. The examples we have like the tetanus vaccine, diphtheria vaccine, botulism vaccine. The characteristics, they are long-lasting immunity. They are often with few doses, generally safe for all individuals including those with weakened immune systems. Require boosters over time to maintain immunity. Then the fifth vaccine, we have mRNA vaccine. These vaccines contain messenger RNA that instructs cells in the body to produce a protein that triggers an immune response. We have, for example, the COVID-19 vaccines. The characteristics, no live virus is used, making these vaccines safer for immune-compromised individuals. Rapid development and production, making them suitable for emerging infections. The mRNA does not stay in the body for long, and the immune response is based on the proteins produced by cells. May require 2 doses and boosters for longer-lasting immunity. Innovative technology with ongoing studies to expand their use for other diseases. Then we have viral vector vaccines. These vaccines use a modified virus to deliver the genetic material of the pathogen into human cells. The modified virus called the vector does not cause disease, but promotes cells to introduce the antigen of interest. For example, we have the Johnson COVID-19 vaccines, Ebola vaccine. The characteristics are one-time doses for 2 doses or 2 doses depending on the vaccine. Can produce stronger immunity response by mimicking natural infection. May cause mild side effects similar to live vaccines. Safe for immunocompromised compromised individuals as long as the vector virus does not cause illness in humans. Our next question is how does herd immunity contribute to disease prevention? First of all, herd immunity refers to a form of indirect protection from infectious diseases that occur when a large percent of the population becomes immune to the disease, either through a vaccination or previous infection. How does it contribute to disease prevention? First of all, reduces disease transmission. When a high proportion of the population is immune to a disease, the pathogen has fewer opportunities to spread from one person to another. This is because there are fewer susceptible individuals for the pathogen to infect. Second, we have protects vulnerable populations. Individuals who cannot be vaccinated, e.g. infants, the elderly, immunocompromised individuals, are on herd immunity for protection even if they are not immune. An overall reduction in disease transmission in the production reduces the likelihood of exposure to the pathogen. Herd immunity helps protect those who are in high risk of several diseases or complications by reducing the chances of them encountering the pathogen. It also breaks the chain of infection. Diseases spread through chains of infection where each infected individual can transmit the pathogen to others. With herd immunity, enough people are immune so that the pathogen is unable to circulate effectively within the population. This makes outbreaks less likely and helps prevent epidemics and pandemics. It also lowers the overall burden of diseases. With a significant portion of the population immune, the overall number of individuals who become sick decreases. This reduces the burden on healthcare systems, preventing hospitals from becoming overwhelmed and ensuring that resources are available for those who need medical care. It also helps to eliminate diseases. In some cases, herd immunity can lead to the elimination or eradication of diseases. This is the ultimate goal for certain diseases. For example, the smallpox was eradicated globally in 1980 due to widespread vaccination leading to herd immunity. We also have the polio is another example, with efforts to vaccinate large numbers of people contributing to a significant decline in global cases and bringing the world closer to eradicating the disease. Our last question is the difference between active and passive immunization. The active immunization is where the body generates its own immune response, like antibodies and memory cells. And for passive, preformed antibodies or immune cells are transferred from an external source. For active, we have long-lasting immunity, often lifelong due to the memory cells. But for the passive, temporary immunity lasting only 2 weeks to months. Again, when we come to the active, takes time, days to weeks for the immune response to develop. But for the passive, immediate protection. May require boosters to maintain long-term immunity for the active, but for the passive, no boosters needed as the immunity is not long-lasting. For active, we have vaccination, e.g. the flu vaccine, MMR, natural infections, e.g. chickenpox. But for the active immunoglobulin infections, monoclonal antibodies, maternal transfer of antibodies needed. Then for the active, it stimulates the immune system to produce antibodies and memorial cells. But for the active, it provides ready-made antibodies and no immune activation. That is all. Thank you.